All right, this podcast is on gene expression. I don't know if you guys know, but there are some rats that are actually genetically programmed to prefer alcohol over water. So it's in their genes that they're going to want alcohol over water. And so if we can understand why rats prefer that, then maybe we can understand why alcoholics have that problem. And so we can get them effective treatment or prevent alcoholism altogether. So in review, so far we have learned that DNA is going to make RNA, that's called transcription, and then RNA is going to make a protein, and that is called translation. When the protein is made, we could be making primary, secondary, tertiary, or quaternary level proteins. What determines that is environmental conditions and the development of the cell. So an example of this is when an extracellular signal is going to bind to the receptor. It sets a signal transduction pathway in motion. And so what happens in the end is that some genes are going to be activated and some genes are going to be repressed. So basically all this is referring to is that we can control gene expression. We can control what genes are turned on and what genes are turned off. Gene expression is modified to counteract changes in the environment to help maintain homeostasis. So if we need that gene to be turned on to help us maintain homeostasis, then that gene will end up being turned on. In other cases, gene expression changes so that the cell can perform a specific function. For example, all of our cells carry the genes to make keratin and hemoglobin, but keratin is only made in specific cells, and hemoglobin is only made in specific cells. So all cells can have the genes, but not all cells are going to express the genes. So basically, if you think about it, there are two types of genes. There are constitutive genes, which are always expressed all the time, always active. And then there are inducible genes. And these are genes that can be turned on and turned off. They're expressed only when their proteins are needed by the cell. So obviously for this chapter, because we're talking about gene expression, we're only going to focus on inducible genes. Genes can be regulated positively and negatively. Positive regulation, it means that there's going to be an activator involved. That activator has to bind, and then it's going to stimulate the transcription of the gene. Negative regulation is there's going to be a binding, but it's not going to be an activator. It's going to be a repressor, and that repressor, after the binding, is going to prevent the transcription of the genes. So activators and repressors are actually called transcription factors. And so we're going to look at how this works in viruses, bacteria, and eukaryotes. Okay, so from your background in biology, you should know that viruses are not living and that they can only reproduce within a host cell. They are not made of cells. However, they can have DNA or RNA. They can have double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. And so basically what viruses do is they take over their hosts so that they can make proteins, and then within minutes that happens of entering their host. There's two types of reproductive cycles that I briefly want to review, and that's the lytic cycle and the lysogenic cycle. The lytic cycle, lytic means break, which means the host cell will be destroyed afterwards. And there's 6 steps. The virus is going to infect the host cell, and the viral DNA is going to enter the host. It uses the bacterium's enzymes, RNA polymerase, to do transcription. One early protein that the virus had genes for is going to shut down bacterial gene transcription. So the bacteria is no longer going to be able to do its own transcription. Another gene from the virus that gets made into a protein is going to stimulate replication. And then another gene that makes a protein that's going to stimulate gene transcription. And then eventually all these proteins of the viruses get made and the protein lyses the host cell. So the virus is able to control, by turning on these genes, to get these proteins made that they need to be able to reproduce. Here's a picture of the lytic cycle. All right, we're going to move on to how bacteria do gene expression and gene regulation, and this is called an operon. An operon is a cluster of genes with a single promoter that codes for proteins in the DNA of bacteria. It codes for basically 3. The specific one we're talking about is going to code for 3 enzymes, and these enzymes metabolize or break down lactose in bacteria. It's called the lac operon. And this is an example of a negative regulation. There are 4 major components to an operon. Component number 1 is the promoter region. This is just a piece of DNA to which the RNA polymerase enzyme is going to attach to start transcription. You can see on the sample diagram that there are actually 2 promoters. One promoter is for the regulatory gene and one promoter is for the structural genes. Component number 2 is the structural genes. These are DNA sequences that code for the several enzymes that we're going to want to make to be able to break down lactose. So these genes are going to be called gene Z, gene Y, and gene A. Each of these genes code for a different enzyme. You can see the names of those down at the bottom. All right, component number 3 is the operator. It is located between the promoter and the structural genes. It is also a piece of the DNA that is able to control RNA polymerase's access to the structural genes. It's like a switch. It can turn the operon, the whole thing, on or the whole thing off. The last component is called the repressor protein. This is a substance that can prevent gene expression because it can bind to the operator, and that will prevent RNA polymerase from transcribing the structural gene. Transcription would resume when the repressor is removed by a molecule called an inducer. All right, so here's some background information. When you drink milk, it is made of a disaccharide called lactose. Lactose, when you drink it, is soon going to be present in your digestive tract and soon in your intestines, and it's going to be available to the bacteria that's living there. Before E. coli can absorb this lactose, it has to have those enzymes. It has to have, specifically, let's say it has to have beta-galactosidase, which is the enzyme that's going to break lactose down into the monosaccharides, glucose, and galactose. All right, so here's a picture of lactose. You can see it's a galactose and a glucose bonded together by the glycosidic linkage. And there are 3 enzymes needed to break that down. Beta-galactoside permease moves the sugar into the cell. Beta-galactosidase hydrolyzes lactose to glucose and galactose. And beta-galactoside transacylase, I'm not sure how to say all these, transfers acetyl groups from the acetyl coenzyme A to certain beta-galactosides. Very unclear. Does it matter? No, you don't need to know the names of these or anything, but it's just the process, knowing that we need these enzymes to be made so that we can break down lactose. Okay, so it is in the bacteria's best interest to focus its energy on using available nutrients. Therefore, E. coli is only going to make the enzyme if lactose is present. If lactose is not present in the intestines, then why would it make the enzyme? It's a waste of energy. So how does E. coli do this? It does it by using and managing the operon. Okay, so here's a picture of the operon when lactose is absent. Here is RNA polymerase. Sorry, my writing is so bad. It wants to go to the operator and to the promoter and be able to go and transcribe these genes Z, Y, and A, which makes those enzymes. However, when lactose is not there, this repressor is going to bind to the operator. When it binds to the operator, RNA polymerase cannot bind, and so therefore it's preventing RNA polymerase from binding to the promoter. It cannot move to the operator, and it cannot do transcription. Transcription ends up being blocked. And that means that these structural genes are not being transcribed and no enzymes will be made. No mRNA is produced, no enzymes produced. There's no lactose, so we don't need to worry about it. It's a way that they can control and manage their energy. All right, if lactose is present, here's our little lactose molecule right there. Lactose is actually going to bind to the repressor. It changes the shape of the repressor. The repressor is no longer able to bind to the operator. So RNA polymerase can bond to the promoter, move along the DNA, and make the enzymes, beta-galactosidase, permease, and translacylase. And then translation's made, we got our enzymes, and everything is good. Okay, so this lac operon is called an inducible system. Allolactose, which is an alternative form of lactose, is the inducer. It leads to the synthesis of enzymes to break down lactose in the lactose metabolizing pathway. It binds to the repressor protein and it prevents its binding to the operator. So now we're going to look at an example that's called the repressible operon, and this is called the Trp or Trp Operon. Okay, so this operon is the Trp Operon, and Trp stands for tryptophan, which is an amino acid. This operon involves a co-repressor. It is a molecule that's going to bind to the repressor, causing it to change shape and bind to the operator, thereby inhibiting transcription. When tryptophan is inadequately present in the cell, it is energy efficient to stop making the enzymes for tryptophan synthesis. So if the cell has enough tryptophan, it's going to stop making the enzyme to make more tryptophan. So it's controlling it. Therefore, tryptophan functions as a co-repressor and binds to the repressor of the Trp operon. This causes the repressor to bind to the Trp operator to prevent transcription. So I know this is very wordy, but let's look at the picture. All right, so we're going to look first if tryptophan is inadequate, there's very low tryptophan, or it's completely absent. So our DNA is going to do transcription and it's going to make our RNA. And RNA is going to have these genes that's going to produce an inactive repressor. This repressor is inactive, cannot bond to the operator. So the RNA polymerase can go along DNA and do transcription of all of these genes and do translation, and we end up getting 5 enzymes, and we can make our tryptophan, which is what we want because we don't have enough tryptophan, this amino acid, in our cells. So everything's going great until now we have too much tryptophan. There's too much present, way too much. So we need to control it. We need to stop. So DNA is going to make our mRNA, and we still have our inactive repressor there. However, tryptophan is present. This tryptophan, which is called a co-repressor, is going to actually bind to the repressor. And when it binds to the repressor becomes active, and now that active repressor can bond to the operator. This prevents RNA polymerase from bonding to the operator, and now we cannot do transcription, we cannot do translation, and that way we do not make the enzyme, so we cannot make any more tryptophan, which is what we don't want anymore. We don't want anymore because it's already there, it's too much. So this is called a repressible system. The product of the pathway, which is the co-repressor, is bonding to the repressor protein, which is then able to bind to the operator and block transcription.